Okay, so what are some diagnostic uses? Now, like I said, I prefaced this before. This is by no means telling you everything that you can use an EKG for. Okay, you got to have years and years of training to understand all of this. This is just some easy examples that you pull out. Simple things like tachy or bradycardia, and whether or not the heart's beating too fast or too slowly. Obviously, that's easy to recognize on here because you've got a calibrated time interval on the bottom, and you can actually measure how frequent those impulses are being sent. Arrhythmia, any sort of abnormal rhythms are going to be evident for sure on these wave patterns that you see. If there's something stuck in between them, that kind of thing. Atrial flutter. In the atrial component, especially at the beginning around the P wave area. If there's any sort of kind of spastic waves that start showing up that are very regular though, or it hits in an irregular fashion before another wavelength starts or whatever. If you can pinpoint to the area on the EKG where this problem is occurring, flutter is sort of like you can imagine what a flutter is, right? But it's regular. Okay, it's not just all over the place with different levels of impulse and infrequent and all that kind of thing. The difference between flutter, for example, and fibrillation is, fibrillation looks like this. You get a bunch of distorted waves and it's just all over the place. Whereas a flutter would be very constant with a whole bunch of waves right after each other. That kind of thing. Okay, so here's atrial and ventricular fibrillation. You can look at that. Now, when I say heart block, I'm not talking about an actual physical substance is blocking blood flow through the heart and that kind of thing. Heart block has to do with the nervous side of things. Whether or not the impulse being sent from, let's say, the SA node through everything else, through the AV node, through the bundle of His, all of that, is that blocked or does that proceed and go all the way through? Heart block is when, a complete heart block anyway, is when the SA node may be firing and the pacemaker cells may be working, but it's not transmitting that message to the rest of the heart through that nerve network. Okay, those cells are not propagating that nerve impulse. That's heart block. Cardiac myopathies, kind of a generic category. Anything wrong with the heart tissue itself. They're not getting fed. There's not enough oxygen. There's not enough circulation. There's coronary atherosclerosis in the arteries that are feeding the heart. This could all lead to just general cardiac myopathies. So that, of course, is not what it would really look like, but it's just an illustration. It would look similar to that. There's a normal rhythm. You can see if they're really close back to back, it's easy to recognize a tachycardic situation. Extrasystole, an extra contraction, in other words, a premature beat. You'll get this sometimes whenever you have like an ectopic focus, or somewhere that the firing goes wrong, and it causes, let's say, in the ventricle region, an impulse to be sent, and it's off timing from the SA node that came sent in. It could be an extra beat, an extrasystole. Fibrillation. And of course, the whole point of defib, right, is to defibrillate something, to shock it and actually reset those electrical potentials. Put enough energy in there to where you actually force these cells to repolarize back to the starting condition in which they were in, and reset this, basically, and reset this electrical activity that's going on here, to defibrillate something. Heart block, you can notice here, you'll see P waves, meaning that the SA node was firing, and it was generating an impulse, but sometimes it was not propagating throughout the rest of that cardiac tissue. So there's one here. There's one here. Nothing happened. Another one, then it did happen. Then another one, and it didn't happen. You get this situation where there's blockage in terms of that transmission. That's heart block. As far as an MI, a heart attack, myocardial infarction. During this infarction, obviously you get sort of like not a tetanus. We can't really go into tetanus, but you get a situation where the cardiac tissue contracts, the muscle tissue contracts and sort of just holds it and sort of, has spasms, sort of. And as a result, your EKG would sort of look like this. And once the ventricle starts to systole and start to contract, then it kind of just plateaus out and fades. That's what this EKG might look like. Okay, these are some easy terms. End systolic and end diastolic volume. Just keep in mind what systole means. That's contraction. Diastolic means relaxation. So when I say end systolic volume, it would make sense then that after a contraction, this is the amount of blood that remains in the heart. Okay, so realize that it's not perfect. Every time your heart beats, it doesn't squeeze out every single little drop of blood that's inside of it. There's always a little bit left over. And that little bit that's left over after a contraction is what we call the end-systolic volume. The end-diastolic volume is after it's relaxed to its fullest, right before it contracts, how much blood is in that. Okay? That's the end-diastolic volume. So your stroke volume then, how much your heart pumps out with each beat, naturally it would make sense that the difference between those two is what your stroke volume is going to be. Okay? Okay. Your cardiac output is how much blood you can pump per minute from your heart. On average, what is it? Do you remember? Around what? No, not quite. That's right. Total cardiac output. Now, I'm not talking about just average stroke volume. Then you can get somewhere like 70 milliliters and that kind of thing. I'm talking about actual volume of blood pumped per minute through your heart, through your circulatory system, out of your heart. Okay, I'll let you look it up. Heart sounds. Yes, 5 liters. Okay. 6. Well, I mean, that's an average. I mean, somewhere around 5 liters. Okay. All right, heart sounds. You should know that heart sounds, when you're listening to that lub-dub sound, obviously it's because of pressure changes associated with the valves that are closing. Okay. The most important thing that's going to create these sounds that you're hearing is a contraction of the ventricle. When the ventricle contracts, what it's going to do is force shut the AV valves, right? The mitral valve, the bicuspid valve, and the tricuspid valve. It's going to force those shut, and that sealing of those and forcing those shut is going to be that first sound that you hear. And then after it squeezes it out and it relaxes, the top valves, the semilunar valves, are going to then close, and that's going to make that second sound that you hear. Okay. So the end diastolic volume, here's some averages to give you an example. This is not the case for everyone. Okay, like we said 70 a minute ago, somewhere around this area. My point is just to show you the math here. If you have an end diastolic volume of, say, 135 milliliters, in other words, that's how much is in your heart prior to pumping it out. Okay? And then it hits and it squeezes out a certain stroke volume. Well, how you can calculate that stroke volume, I mean, you can't just pull it out of somebody and collect it one heartbeat. It's not very feasible. But if you could figure out a way to calculate the end systolic volume, in other words, here it is relaxed, it squeezes out a certain amount, maybe we don't know what it is, but once it's squeezed out, if we know how much is left behind, then obviously we know the difference between this and this has to be our stroke volume. Okay? All right, as far as, like I talked about heart sounds, and that kind of stuff, you've heard of heart murmurs and all that. Sometimes they're hard to hear. Sometimes if they're really bad, of course, they're easy. You've got stenotic, you've got insufficient valves. Insufficient valves are sort of like, you've heard of mitral valve prolapse, perhaps. Something prolapses, it turns back in on itself. So like in the case of mitral valve prolapse in your left ventricle, between your left atrium and your left ventricle, what will happen is some of the blood from the left ventricle can leak back into, because that mitral valve prolapses. Here's your left ventricle. It's supposed to hit, squirt through that valve, and then when it fills up the left atrium, my bad. Here's your left atrium. It's supposed to hit, squirt it through that mitral valve into your left ventricle, and then as that builds, it's supposed to push back against and close that valve. Okay? If you've got an insufficient valve, and worst case scenario, an insufficient valve leads to what's called prolapse, that can fold back in, and some of that blood can leak back. Okay? And if it does, that obviously the ramifications of that are you get less efficient blood flow. You're not pumping out as much as you could. You're not doing. The whole purpose of a 4-chambered heart like we have is to most efficiently separate the good and the bad blood in terms of the oxygenated and the deoxygenated blood. The more leaking that you have and the more backwash you have, the less efficient that separation becomes, and you get less oxygen delivery to the body. Your blood is not as high in content of oxygen as it should be. That kind of stuff. So with mitral valve prolapse, one of the things is you may get winded a little easier, you might get tired a little easier, but that's in severe cases. Anyways, as far as murmurs go, murmurs are just these weird sounds that you can hear with a stethoscope. Because of these valves, say you have an insufficient valve that could lead to what's called mitral valve prolapse. How would insufficiency sound in terms of a murmur? You would hear a whooshing sound as you were listening through the stethoscope. If you had a stenotic heart murmur. Stenotic valve. Okay, that means it's sort of gotten rigid and it's very fixed. And instead of the opposite, instead of being too loose where it can kind of flap around, it's very rigid. So it's like blowing across the top of a pop bottle. It actually whistles as the blood comes rushing by. So you can infer that from the sounds that you hear. Okay. I mentioned rheumatic fever. Some of these cardiac myopathies, actually stenotic valves. Some bacteria have been shown to able to get into the heart and actually deteriorate these valves, and as so, stenosis comes into play with rheumatic fever and that kind of thing. You can research that on your own. You don't have to know it for this class. But I just mentioned. So stenosis, stenotic murmur is a whistling sound. Insufficient murmur is a swishy sound. If you look at page 325, now that's from the old book. I don't know what it is in the new book. It's a table that has all the heart murmur sounds on it. What's that? Yeah. 319 it looks like in the new book. I don't know how much experience you guys will have with this. I don't know if you'll ever have to, I don't know what the detail of your job description is. But, if you just want to play around with it, if you get a situation, if you're working with someone and a physician or whatever, and these murmurs come into play, maybe you want to listen to somebody's and get an idea of how this works and hear the same thing there for your own education. This is something you might want to keep in mind. You might want to look at this table, keep it for a reference. It helps you identify. If you're listening to the lub-dub, realize that the lub is going to be an AV valve, right? It's the first valve that closes between the atria and the ventricle. The dub is going to be due to a closing of the semilunar valves. So if you can hear, for example, lub-dub whistle, you can pinpoint where this is. You can get an idea. Now, as far as the left and the right side of the heart, that's harder. That takes a lot more training. But in terms of just whether it's on the top half of the heart, in terms of is it a semilunar valve or is it an AV valve, you can certainly listen and figure that out. Listen to the heartbeat. Is it a lub-dub whoosh? Is it a lub-dub a whistle? Is it a lub-whistle dub? These things will tell you different things, and you can see that on that chart. Because if you know that if you hear a lub sound followed by a dub sound, then a whistling sound, you can logically deduce which valve it must be based upon how you know the blood travels through the heart. Okay, so you can take a look at that and see if you can make some sense out of it. And if you can't, if you've got some questions about that, ask me. But I'm not going to stop and lecture on it. Figure it out. Okay, now I mentioned that the cardiac tissue is self-excitable, and that's very important. It auto-regulates itself. We have pacemaker cells. We have that drift and fire mechanism that I talked about because of the slowly closing potassium gates and the ever-leaking sodium gates that eventually causes a rise in potential to trigger some transient calcium gates, and before long you've got an action potential, and it does the same thing over again, and you've got this distance in between the two in terms of time. That is your heartbeat. Okay? So it is self-excitable. You could pull a heart out of a human body and hold it there, so long as it's got blood to let the tissue survive in. It'll survive on its own for a little while. Pull it out, and you can hold it in your hand, disconnected from a nerve supply, and it'll sit there and beat in your hand because these cells have the ability to do that. However, realize that when it's wired into your body, Indiana Jones, pull it out. When it's wired into your body, though, realize that it's now just not its own organ. It is part of your entire homeostatic makeup that has to make you you and to get you to function in the environment in which you live. So as such, when it is hardwired with nerves, those nerves, based upon feeds from all other parts of your body, can have an influence on your heart rate. It can override the autorhythmicity of the heart. If, for some example, epinephrine and norepinephrine get released, and you have a fight or flight type of situation, your blood pressure, is going to shoot up. Your heart rate is going to shoot up. It'll override these autonomic nervous system. You don't have to think about it. The autonomic nervous system will feed into the heart and cause it to go faster than normally what it would. And then when that subsides, you're back to your pacemaker cells again. So they are not directly responsible and ultimately control all of the heart. It is actually myogenic for the most part. The heart, it'll control itself, but it can have neural feeds to override that which is happening. Okay? And this is where we get into what's called sympathetic and parasympathetic divisions of the autonomic nervous system. Okay? If you think of, now, it gets a little tricky. I know when I get into digestion, we're going to have some more practice with this, but when I start talking about sympathetic, parasympathetic in terms of what's what. How do you know what happens? Do the blood vessels relax? Do they dilate? Do they constrict? Does the heart rate speed up? Does the heart rate slow down? What happens to respiration? And how do I know whether it's sympathetic or parasympathetic? Because it's easy to get them all confused. Just think about what's happening in the body. Is whatever is happening sympathetic to the situation to allow you to survive better, or is it not? So for example, and that's a weak explanation for right now, but I'll give you more when we get into digestion. But it's easy for the heart. It would make sense that sympathetic activity from your autonomic nervous system is going to stimulate and help, like if it's a fight or flight response especially. Something to be sympathetic to that stimulation would be something that could increase your heart rate in response to that. Okay? So sympathetic activity is going to increase your heart rate. Parasympathetic activity is actually going to work against that and work to bring that back down in the opposite direction. Now what you will see is it gets more tricky trickier, whatever the word is, as we look at some other systems. But like I said, for the heart, it's pretty simple. Sometimes, however, you will see, like in the case of the blood vessels, and we're not going to be talking about the blood vessels, but they only respond to sympathetic activity. There is no parasympathetic innervation in your blood vessels. So it's not a matter of constriction and then parasympathetic comes in to relax it. It actually is just the degree of sympathetic stimulation. Either you get a lot or you get a little. That's it. It either comes on or it lets off, but there's nothing that actually, like inhibitory, that fires against it to try to shut it down. Okay? But there is in the heart. Okay, your control of stroke volume. Now, this is another thing that we can do, is control how much blood is pumped out with every heartbeat that we have. And it has to do with how full our heart can get. Now, realize when I say how full, especially like the left ventricle. That's the meatiest part of your heart. If you look at the tissue and the thickness, your left ventricle is the thickest chamber of your heart because it has to pump against what's the biggest afterload. We call it the afterload. It's the amount of blood and the pressure. You know that your aorta, right, has a tremendous amount of pressure. If you stabbed yourself in the aorta, it would shoot for several meters. Actually, it would just be this arch of blood streaming out of you. There's a high-pressure situation in the aorta. Since it's confined into the vessels, that pressure then is backed into the heart. So your left ventricle has to pump against that pressure. So you can imagine that as heart disease sets in, atherosclerosis, vessels start narrowing, more increased blood pressure in the peripheral areas in general, they work against that afterload. It brings even more pressure back against the left ventricle, and it's almost a feed-forward mechanism. It's almost like a positive feedback type of cycle because the more the pressure is, the harder the heart has to work. The harder the heart has to work, the worse off it is. Okay, and it degenerates, and it keeps getting worse. It's progressive. Anyways, our heart is set up in a way that I talked about resting length, optimal resting length. The best situation where you can have actin positioned next to myosin to give them the maximum amount of interaction to get the best contraction that you can get. It just so happens that our heart is designed so that as it is resting, okay, after it hits and as it is just sitting there, say they're pretty close together, it is not at optimal length. It could contract, but if it did, it would only give you a little bit of a stroke volume. It wouldn't give you that much. What our heart is made to do, it's called the Frank-Starling law. What it is made to do is as it fills with blood and the tissue gets bigger, as it spreads out, it actually reaches optimum length. So it is made to expand in order to reach optimum length, which is good. Because what we actually see then in the heart is the fuller it gets, the closer to optimum length it gets, and the more efficient it is. Okay, so the more blood it holds, the more contraction it can get. To a certain degree. Obviously, if you engorge it and increase the pressure to the point where you overextend it, that's a whole different issue. That's a problem. But to a degree, you pull it out to reach optimum length so you can get a maximum contraction and force out a bigger stroke volume. Okay. That's what this is saying. So. Okay, like I said, high blood pressure, why it's a problem, is mostly because of the toll that it takes on your heart. It increases the amount of afterload, the amount of back pressure against what the left ventricle is trying to squirt out. Okay? And the harder that is, the harder it has to work. Let's see. That's what this is saying. So, now, what that could mean is a lack of good blood flow in general, because the whole point, obviously, is to take nutrients, to take hormones, to take cells, to take whether it's white blood cells, red blood cells, to take oxygen and distribute it all throughout the tissues, all throughout your body. if you're working against a large afterload, okay, a large pressure coming back onto the ventricle, you can see where it's sort of like trying to row upstream. Okay, your body is not going to be as efficient, nearly as efficient at delivering all those things I just said as it needs to be. It's going to be slower, and by the time it spreads around, it's not going to have been as efficient. Things aren't going to be, not as much would be delivered as normally, and that kind of thing. So when we use the word heart failure, this gets me every time. And people, you know more about this than most people, obviously, because of the profession that you all are in. But I hear the word a lot: congestive heart failure. What was wrong with him? Why did he die? Whatever. Congestive heart failure. Well, there's a lot more to that. Congestive heart failure is a catch-all term. It's like a default term because it's very generic. Congestive heart failure just means that there was some sort of load placed on the heart for who knows what reasons, and it was failing to pump out the amount of material and oxygen that it needed to meet the body's demands. So what does that tell you? Really nothing, because we don't know what it was that caused that, what the origin of that situation was. There's a lot of things. So it's sort of like what you say if you really don't know, in terms of something was wrong with the heart, but I don't really know what happened. So anyways, anytime you have just a generic situation where cardiac output is failing to meet the nutritional demands and the removal of waste in your body, we call that heart failure. Okay, like I said, there's a lot of things that could lead into that. There's a lot of different ways you could have heart failure, but that's just a generic term. The most common causes, we've got damage to heart muscle. Say like a heart attack, a myocardial infarction. You've got reduced blood flow to a region of cardiac tissue on the actual coronary arteries that will not allow blood to go where it needs to go, and as such, oxygen doesn't get to where it needs to go, waste removal cannot happen, and these cells in the actual heart tissue start to die. And when they start to die, the heart itself obviously loses contractility as the contractile cells start to die. That's a heart attack, is when you have this necrosis of tissue, this death of some heart tissue. So that's one way you could get damaged heart muscle. Prolonged pumping against a chronically increased afterload. You can get atrophy of the heart, not atrophy, hypertrophy of the heart as a result of that. Just like muscles that are overworked, they get bigger, right? If you keep having a chronic afterload, you can actually get the heart to grow bigger. That's not always a good thing. That's not like, you don't want beach muscle heart, okay? You have a certain size that you need and everything like that. It's detrimental to try to grow the size of your heart. Okay, so ultimately what ends up happening is things get sort of more rigid, not as flexible, not as smooth. So ultimately what happens is the stroke volume decreases with cardiac conditions, and that just means blood's not getting pumped out like it should. Now, something to keep in mind about coronary arterial flow. Coronaries are the tiny little arteries. As soon as you come out of the left ventricle and you go past the semilunar valve, okay, coming out of the left ventricle going up into the aorta, right on the almost trapped inside about where that valve opens, there's little sinuses in there that feed into the coronaries that are actually it's a high pressure situation, so that pressure drives that blood down into those little openings. And those openings take the blood actually out to the heart and feed the heart tissue itself. Those are the coronary arteries. When you see the picture, it's those vessels that run on the front of the heart. Okay, obviously very, very, very important because that's feeding your ticker. And if your ticker don't tick, then we got a problem. So you've got to have those arteries there. But what's important to note is most of the blood flow through that area is occurs during relaxation, during diastole of the heart. And it makes sense if you think about why. Because like I just said, when that heart contracts and it opens up those valves, it's got this momentum and it's taking that blood past that up into the aorta. Now when it relaxes and some of that blood can settle back a little bit and those valves are closed, it's easier for that stuff to drain out into those coronary arteries. So it's actually compressed. Those arteries become compressed during the contraction, during systole. So, which is also realized too. What happens in like the case of fibrillation or a myocardial infarct, and you got like a sustained contraction. Keep that in mind too, that you're compressing coronary artery flow when you do that. So you're adding a double whammy to what's going on here. Okay, how does coronary blood flow change? How do you get more? How do you get less? That kind of thing. Primarily in response to your heart's oxygen requirements, and that makes sense. But there's a couple ways that we can go about that. So it says the link that coordinates coronary blood flow with myocardial oxygen needs is adenosine, the same stuff that's in adenosine triphosphate, ATP. Where there's ATP breakdown and usage, there will also be adenosine floating around. Okay? So when oxygen is low, when there's a low actual partial pressure, what's called partial pressure of oxygen floating around in the blood, or if there's more ATP being used as an energy source, if it's cranking up the amount of ATP it's using, that will cause more. It would cause coronary dilation is what would happen. There would be a second messenger system kicked in by the presence of this adenosine that would the events would turn into a situation where dilation would occur and more blood flow would occur. That's what that's saying there. And there's what I just said in flowchart form, so you can look at that yourself. Adenosine's the key. All right. Now, this is interesting because this is highly debatable among many people, and again, beyond people who know way more about it than I do. But atherosclerosis, we think about, and I even say it sometimes, even though I know it's wrong. You're just, you got those Rally's fries, right? I mean, seriously, they're good. And I'll tell you what's best. By the way, if you're going to Rally's, is the number. I don't know if it's number 4 or number 6, but it's the one with it's got two hamburger patties, cheese, barbecue sauce, onion rings on the hamburger. Oh, God bless that sandwich. Bacon. It is awesome. Anyways, speaking of atherosclerosis, it is not like you're just eating these saturated fats and these high LDL type of and all these different whatever words you want to pull out. It's not like you're eating all these and they're just going down into your vessels and they're just lodging up there and they're clogging up everything. That's not quite the way it works. You actually have these plaques that form, but it's not because things get snagged and they block up because of, you know that when you eat stuff, it passes through your digestive system and it breaks it down into tiny little molecules. So it's not like you're just injecting this fat straight into your veins or anything. So actually, you can go so far as to say the cause is unknown in terms of atherosclerosis. Now, there is a really neat thing I'd like for you to read. It's in your book. It's a little article, a little excerpt, separation, I don't know what you would call it, about atherosclerosis. I'm not sure what page it's on. Keep flipping. Yeah, page 330 in the new book. I'm not sure what it is in the older book. But you can't miss it. It's huge. I think given what you're going to do and the field that you're in, this is something everyone needs to know a little bit about. Plus, just in general health, you should know this about your body. Get an idea as to what theories are out there that they suggest might cause atherosclerosis. And bottom line, what you'll see is really the only thing that they can agree on is some sort of almost like an immune response. Because you hear it all the time that smoking can increase your risks of atherosclerosis. Diabetes can, being overweight can, eating unhealthily can increase your risk factors. And yet some people don't do any of those things and still die still get atherosclerotic plaques form in their body. Now that is by no means an excuse to just live a horrible lifestyle. I'm not saying that. I'm just saying there's more thought. It's easy to point the finger, but realize there's a whole lot of stuff that goes into it. And certainly all those mixed together. Genetics could certainly be a factor, but that's not ever going to be the only factor. I mean, everything comes in, so it's a very complicated thing to try to explain in terms of what the cause is because there's so many different ones and how they interplay depending on the situation. What basically happens, though, the most accepted theory, I'm not going to go into the details, I'll let you read it, but when you look at this picture, okay, you have very smooth interior linings of your blood vessels for obvious reasons. You want things to have little friction and for things to slide through very easily. These plaques that form, this is atherosclerosis, this scarring of this fibrous tissue that forms, that's sclerosis. Okay, this tissue that actually forms just beneath the endothelium of the blood vessel, not out into the blood vessel itself. It actually wedges itself in between the tissue that makes up the blood vessel. And as this plaque continues to grow, it narrows this opening. Okay, you can hear things of like if somebody heard you 80% blocked or 70% blocked. We're talking about this narrowing. Not only when you get an increased afterload and with other cardiac conditions, this all feeds into other things because not only will these plaques continue to grow, but when they do, you lose elasticity of this blood vessel. So the more of this you have, what else you've got to deal with is the fact that it's not being able to expand to accommodate pulsations and different kinds of things that are pushing this blood through. So when it's blocked, it's actually very rigid, very tough, and it starts to get more and more occluded. It's not like you can go in there and just do a little scraping like they do at the dentist office and get rid of it. That's not the way this works. What actually ends up happening is, for some reason, this lining probably gets irritated. This is why smoking could lead to the same thing, but it would depend on the person and how predisposed they are to getting irritated in terms of the blood vessels, not in terms of their personality. But so as you come in here, something happens to where these linings start to get irritated to the point where macrophages are attracted to this area. Macrophages are like the generic type of white blood cells, right? And one of the things that we talked about was macrophages, when they get somewhere, they like to stay there. They come in and they become what's called resident macrophages, and they stay there and they become part of that tissue. That's what's going to lead to a lot of this, because what happens is when those macrophages especially come in here and start to react with whatever's going on with this blood vessel, you'll also see that cholesterol does have an important factor, especially your LDL cholesterol. I'll talk about the different types in a second. But cholesterol molecules are important. They're not bad. The problem is whenever they get brought in and instead of going to do what they're supposed to do, get swallowed by the macrophages. And these macrophages are trash takers, right? They soak up everything and they start to engorge themselves with these type of molecules. And when they start to die, there's even more that can go on. When these macrophages start to die, there's some chemical reasons, without trying to lose you, there's some chemical reasons associated with the products when they've stored them. Once they die and start to get released, it sort of destroys this even more. So it's, like I said, it's a feed-forward type of situation once it starts. And the bottom line is this plaque continues to grow on the inside. It's waste and debris and scar tissue and everything else from this type of reaction. So eventually it could close, worst case scenario. But the whole time, if this is localized, that's one thing. But if you are a person who's, let's say there's something with your cells, there's something genetic about your cells that predisposes them to be irritated more easily than someone else's. In which case, smoking is certainly going to increase your risk. Lack of exercise is certainly going to increase your risk much more than a normal person. And there's no way of knowing that because there's no test out there that automatically just give you that magic answer. But anyways, all of this could lead to an increased afterload in the heart, in which case you could lead up to heart disease. Okay? So anyways, there's a lot that goes into it. I'd like for you to read the theories that are there. Okay, so coronary artery disease is a result of kind of this kind of stuff that we see. Whenever you get widespread atherosclerosis, you get damage to these types of vessels, and where they become most potentially harmful is not just in the body, but in the coronary arteries that are on the heart. Because if those get constricted, we've got a big problem. Okay? You can live a little while with restricted blood flow to your body, but not to your heart and not to your brain. All right. So coronary artery disease can cause myocardial ischemia, which is just a generic term for low oxygen delivery. You'll hear this word a lot. If you ever work in a cardiac unit, there's some catchwords probably that you're going to hear. You know you're going to see the EKG thing all the time. You're going to hear a lot of heart failure, heart disease, MIs, all this kind of stuff. If you hear this word, I just say it down here. If you hear this word, realize all it is a term that means low oxygen delivery, okay, to the actual coronary, through the coronary arteries to the heart tissue. That's what it means. So again, it's generic. You don't know. There are a lot of things that could cause that. But if that is happening, if it's oxygen, it's an insufficiency to this region. That's what this word means. Okay. What could lead to the cardiac ischemia is profound vascular spasms associated with the atherosclerotic plaques that we just looked at. Spasms, of course, things get screwed up, things get shorted out in terms of the circuitry of the nervous system. Those can start constricting on their own, maybe as protective measures, who knows what. Spasms start happening. That's going to restrict blood flow. That's going to decrease the amount of oxygen bringing to the cardiac tissue. The atherosclerotic plaques that I just talked about leads to occlusion. Occlusion is a fancy word for blocking, right? So thromboembolism, we know that thromboembolism is thrombocytes are like cell fragments that are involved in clotting, right? An embolus is something that's moving through. You could have a gas bubble that's an embolism. A gas bubble could be a little pocket of air that's floating through here and block up this little vessel right here, and that could be an embolus. When we say a thromboembolus or thromboembolism, you've got platelet aggregation, you've got some sort of blood components that are actually aggregating here that then can break off and move through the body. Okay, we call that a blood clot, but more specifically a thromboembolism. Now, realize this is not just like a big giant scab that forms on the inside of the body that then sort of like those when the scab starts to form and it's kind of loose and gooey, but you can pull it off and all the blood still stays together, but it's not crusty yet. It's kind of like one of those scabs. You guys don't know? When you tell me you don't pick your scabs, come on. Oh, that's the best, man. All right. You get those cool scars. Anyways. No, because I did it when I was little, not when I was big. So, all right. This should be common sense, but we'll show you anyways. Of course, you know that everybody's heart's a little bit different, right? When I show you this map, that doesn't mean that on your heart, this is where your main coronary artery is going to be. And the number of branches off of it obviously varies with each person. I don't remember the stat. It was interesting. I might have mentioned it to you before, but there are the anatomical terms that you learn and where things are located in people's body. And you learn all that with 101 and 102 and all that. Realize that only applies to like 70-some% of the population. There's actually over 20% of the population has a different layout than what's in the books that you're actually reading, which could explain some things. But sometimes we operate under that assumption that my appendix is hurting, but maybe your appendix isn't even there. Maybe your appendix is on the other side. I don't know. Now it's not a problem so long as there are severe cases of that when everything's sort of like turned around on the inside in people and they never even know. Because if they don't have any health problems and if everything is wired, even if it's in reverse, as long as it's all the same, no one will ever know the difference. A lot of people have connected kidneys, like a horseshoe kidney that's all together. Some people have like 3 kidneys or floating kidneys. You'll never know until that day you go in to take an X-ray or MRI or whatever because you're having some problems, and all of a sudden you've learned that after 30, 35 years, 40 years, whatever, that you had a 3rd kidney that you never knew about. So who knows? Anyways, on this picture, obviously where that thromboembolism lodges itself determines how severe the consequences are, right? If it lodges itself in this, like where A is, you can see that everything after A is going to be affected. Look at all the entire left side of the heart. That's death. Whereas if you get it way down here in the smaller regions like B, you're only losing a little bit of tissue down here. So obviously that's of utmost importance. Cholesterol. We hear it. We think it's evil. And there's all the medications out there because I'll tell you what's funny is these stupid commercials that they have for all these pharmaceutical products. I've never met so many happy people that were so happy that they had herpes in my entire life when you watch these shows. You know what I mean? And like whenever they're talking about or whatever those statins are, whatever I forget which particular one it was. Those medical students are sitting around, they're discussing the effects. They don't do that, okay? I've not went to medical school, but I researched in the med center around all with the med students. So I mean, that is not, they don't sit around and have memorized all the consequences of all this particular pill, and that's not what they're talking about. And when they leave the building to go to lunch, they're not still talking about it, and they're not still having their clipboards, and it's ridiculous. But anyways, the effects of cholesterol and atherosclerosis. Okay, but that seriously.
That's bad.
But anyways, where do we get our cholesterol from? We make a lot of it, and you will see that when we get into hormones, it's very, very important. Cholesterols are precursors to a lot of sex hormones, estrogens and testosterone. We need cholesterol to make those. So we do have self-manufacturing. Also, think all the way back to one of the first chapters that we did when we talked about the structure of the cell. Okay, and we had the plasma membrane, and we looked at the channels that were in it and the phospholipids that were floating around. One of the things you'll remember is that there were little tiny cholesterol molecules sandwiched in between all those phospholipids. So they do have a very important structural role in the cell. So these are not all bad. This is something that we need. It's a matter of when they get into the wrong place and we overload ourselves on it, just like anything. We can't overdo it. And obviously dietary intake. Okay, so we're going to get a lot of cholesterol. Now, here's what, and maybe you already know this, but if you don't, I would like for you as nurses to be able to tell this to somebody, is the difference between, what's the difference between my good and my bad cholesterol? What's the difference between HDL and LDL? First of all, the name. Let's figure out what specifically is different about them, and then why one is good and one is bad. Not really bad or good, but relatively speaking. HDL stands for high-density lipoproteins. Cholesterol is made of pretty much two things: lipid molecule and protein molecules. Okay? Now, they combine together, and depending on how much of each there is, whether there's more lipids or more proteins that make them up, it helps us classify them in terms of HDL or LDL. Protein, well, think about fat and muscle. Which is more dense, do you think, fat or muscle? Muscle. So keep that in mind. High-density lipoprotein has more protein, just like muscle has more protein, right? High-density lipoprotein has more protein compared to the lipids that are making up that little molecule. Okay, so there's a higher percentage of proteins present than there are lipids. That's HDL. That's high-density lipoproteins. LDL stands for low-density lipoprotein, so naturally it has more lipids than it does proteins that make it up. Now, why are these considered good? Why are these considered bad? It's not just because of protein and lipid content. That's just how we identify them. What makes them good and bad is the fact that we say HDL's role is considered good, I guess, because it returns cholesterol to the liver. Okay? Your liver gets to decide, how much, and monitor. It's the gatekeeper of the blood, right? It monitors its levels, it pulls stuff in, lets stuff out when it needs to go. What HDL does is gather cholesterol from your body and is the molecule that's responsible for bringing cholesterol back to the liver. LDL does the opposite. LDL takes if the liver decides there needs to be more cholesterol or whatever, LDL is the molecule that will take cholesterol, transport it out to the body tissues. So you can see why we gave, especially in terms of health, why we consider one good and one bad. Really, they're both fine. It's just a matter of we obviously would rather pull it away from our body, given our lifestyles and the things that we commonly eat as a society. We're better off bringing stuff into the liver and then turning our liver to fat, as opposed to taking it out. You guys ever watch that, what's it called, Super Size Me or whatever? If you've never watched it, you got to watch it. It's that guy who goes on a McDonald's diet for 30 days or whatever. Yeah, and he continually gets health checks and watches his life deteriorate before his health deteriorate before his eyes. You should watch it. It's probably in the video store somewhere. Okay, so obviously LDL, since it goes out to the body, realize one place it can go is to those areas where those atherosclerotic plaques are forming. Okay, so they can obviously contribute in that manner. Now, how does this play out in terms of health? Smoking. How does smoking contribute to heart disease? That kind of thing. Well, there's actually several different reasons. One is, in the case of smoking, realize the only thing that you're sucking in is not just nicotine, right? There's thousands of horrible chemicals that are inside cigarettes. But judgment laid aside, I'm just saying that those chemicals, some of the stuff is actually benzene, an organic solvent found in gasoline. I mean, you've got all kinds of crazy stuff you're putting in. But as that goes in, realize that when those chemicals get dissolved in the blood, if you are already prone or if atherosclerosis is something that's happening in your body, and you wouldn't necessarily know about it, right? I mean, there is obviously a genetic factor because you can trace it through family history and that kind of thing, but you can never be sure. You never know that if in your body, that is going to add to the irritation that's already happening around those atherosclerotic areas. That's one thing that could certainly happen. But also what can happen is it can take your HDL, and this is what this one points out, it can take your HDL that normally is returning the cholesterol, right, back from your body, back into your liver. It can lower those levels. So in other words, you could get to the point where you're having more cholesterol taken out from your liver than you are being returned to the liver, and then therein lies the problem. Exercise promotes HDL. Again, the mechanisms by which this occurs, realize, don't ask me to explain to you how does exercise. They don't really know. There's just correlational studies that have been done saying that they'll let people exercise, they'll measure their HDL. There may be some theories out there that have come about since then. I don't know. But a lot of this is sort of, we see it, but we don't exactly sure why, and that's why they're studying it all. But exercise does seem to promote HDL. Okay? So obviously, maybe you're thinking, well, if I smoke and exercise, I can cancel it out. Smoke a cigarette while I'm on the treadmill, everything's fine. I don't know. My wife can talk better about this. She just wrote a pretty good paper on hormone replacement therapy and that kind of stuff. And if I have time, maybe I'll get her to come in and talk to you guys at the end of the semester. I don't know if she'll be willing to or not. But estrogen has been shown in premenopausal women to actually raise HDL levels. Okay, and that would be obviously a good thing. But there's also some other things. And like I said, this is out. She studied this, not me. Some other things about the different options that exist and how to take the hormone replacement. What's good and what's bad. What are the new things out and that kind of thing. And, it's interesting. Vitamin E and C have been shown to slow plaque deposition, but again, that's kind of ambiguous. There's a lot of unknown there. Intake of saturated fats. We all know, remember the difference between saturated and unsaturated fats from a chemical level. If we think back, saturated fats, like a phospholipid, has that glycerol head. A phospholipid had a glycerol center with a phosphate head and two fatty acid tails hanging down. A lipid, a saturated fat, for example, would be like some of those fatty acid chains that are down here that are completely full of hydrogens attached to those carbons. That means more energy associated with them, basically. And if you have a high intake of saturated fats and don't burn off that energy gets stored somewhere. Okay? An intake of saturated fats increases lipid content. Okay? Now, if you increase lipid content, your body has to react to maintain homeostasis. So what it's going to do is divvy out those lipids and stick them different places. Okay? And sometimes, for some of us, it's the same places over and over. But it'll take that out, and in order to do that, it's got to take that cholesterol and those lipids and form the LDLs that are going to take it away from the liver out to the body. So obviously saturated fats, through the production of lipids and then thus the transfer of those lipids out through the body, that's the link to LDL in terms of saturated fats. All right, so it's not exciting, good news kind of stuff, but it's stuff that everybody should know. Any questions? You know everything. Good. Got through that faster than I thought.